Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about peripheral nervous system. We want to have an overview of the central nervous system, and then to characterize the peripheral nervous system. We want to characterize the neurons and their support cells. We want to look at organization of peripheral nerves in general. If this is useful, please share it with your friends and colleagues. Thank you. The peripheral nervous system. Hi, I'm Larry Johnson. I am a professor at Texas A&M University, and today we're going to talk about the peripheral nervous system. Here we see a host of cells. There's a nerve cell, a epithelial cell. We can see some myelinated axons here, cell bodies here and there, and a little bit about the synapses, and whenever it's synapse associated with a muscle. And we're going to talk about some receptor type nerve endings. So there's four basic types of tissue, and nerve is one of those types of tissue. We see the nerve, of course, in the brain that we see on these two individuals. And here is a nerve cell body right here. There's this nucleus, and the nucleus may look like a snake eye, with a nucleolus in the middle and a granular substance on the outside of the cell body and satellite cells outside. So this is nervous tissue. And so today we're going to talk about an overview of the central nervous system, characteristics of the peripheral nervous system, just a little bit about the central. And we're going to characterize neurons and support cells and organization of the peripheral nervous system. And here we see a host of different types of cells. We can see where one axon is being supported by multiple Schwann cells, and there we can see some axons kind of in yellow here in this scanning electron micrograph with a myelin sheath around each one. Now, why did we scare ourselves? I don't know. This is on a roller coaster. But whenever you think you have a sensation of falling, you release Efferent, and that kind of gives you a feeling that you don't get if you don't put yourself in a dangerous position. Nerve is like epithelium. They both come from the ectoderm. Epithelium comes from more than ectoderm, but ectoderm is one of the sources, is where the nerve comes from. And it also has kind of like a base, like this is a nerve cell body with the dendrites projecting off the surface. There's a cell body here, and then it has an apex, and the apex is kind of like this. This is the axon. So this is where it receives message, the nerve cell, and then where it sends out neurotransmitters to be transmitted at the very end to the target cell. So nervous tissue is specialized for the transmission, reception, and integration of electrical signals. That's what it does. It transmits, allows you to move. It receives, it allows you to feel. And it interprets the images to know how fast you need to run, for example. Now, distinguishing features of the neurons is that they're long, excitable cells that can stretch from the brain all the way down to different lengths of the body, from the spinal cord to different places. So they're very long, excitable, and they transmit their message through a synapse. Synapse is where one cell interacts with another cell, and it does that by chemical signals that are transmitted, like from the nervous cells to the muscle cells, to get them to contract. So transmission, you need something long to facilitate that. You need a way to get signals through fast, and we'll talk about the myelin sheath to facilitate that. Reception, you need some kind of receptors to be able to feel or taste or smell or hear. And then you also need integration, somehow to integrate it so know what to do about it once you get that signal. Now, glial cells are support cells of the nervous system, so they support cells that we see. Nerves are collections of neuronal processes bound together with connective tissue. So a nerve has got connective tissue surrounding the neuronal processes, which are the axons or the dendrites, and the myelin sheath that goes around it. Axons can be coated with myelin, and that would be myelinated axons. That's what we see right here. This is the axon right there, kind of in gray, that we can see. There can see where one's pulled apart there. And then you got the myelin sheath on the outside. Sometimes you have unmyelinated axons. I don't really see some of these. Maybe these are some unmyelinated axons, I'm not sure. But certainly these are myelinated axons for sure. Now, distinguishing features of the central nervous system, you have the central nervous system and the brain and the spinal cord, and then you have ganglia throughout the body. And so the signal comes from the central nervous system to those ganglia, and then those ganglia send a message within that organ that you might be innervated with. So inside of the organs, you have cell bodies which receive message from the central nervous system, and then they kind of control the function of that organ from a local electrical impulses that you're getting there. So the function of our system, in one word, is communication. You step on a tack, you pull your foot up, that has already gone to the spinal cord and back to tell your foot to respond to that. So you get the sensory up, motor coming down to respond to that. So specialized signals and neurons, long process, some of them may be a meter in length. You got the cell body, the nucleus, the dendrites where it receives message, and the axons where it transmits the message. Now, one characteristic of neurons is they're irritable. Protoplasm is capable of reacting to a lot of different chemical and pressure, physical characteristics, and the conductivity. They're able to transmit things long distances. And so those are two important things. One is it can be excitable, and two is it can transmit. And here we see different types of nerves. You got the unipolar nerve. This is a cell body with one projection. You got the bipolar with two projections. And this is what we have in a dorsal root ganglion. Sorry, you don't have a dorsal. This is what we have in a dorsal root ganglia. It has two projections, but it also has this little piece right here, which makes it a pseudo unipolar. So this is what's in a dorsal root ganglion. And then you have the multipolar. So multipolar as well. So this is a dorsal root ganglion. This actually is in the ear that we find there. So the central nervous system, the brain and spinal cord, which are the neurons, which are the cell bodies with the axons and dendrites, and the support neuroglia that are located brain, spinal cord. And then the peripheral nervous system is everything outside the spinal cord that you have and outside the brain. So here we see the dorsal root. These are the pseudo bipolar cells that we talked about. They receive impulses from here. It goes back to the brain, and then it tells your muscle to jerk away. It goes back to the spinal cord, rather. And so this is a spinal cord is controlling this process, and inside the spinal cord you have neuron cell bodies too. So you have ganglion on the outside, you have neuronal cell bodies inside the spinal cord as well as the brain. And so different things from the spinal cord, it interacts with the ganglion cells that are located in the individual tissue to cause contractions, to cause sweat release, whatever. And so the activity of the nervous system is to receive, and that's your afferent pathway. You feel pain, and the process, centralization, is paramount, so that the right arm knows what the left arm does, and your foot and legs know what to do. So whenever you're running, you're using your arms, even though you're swinging your arms to help balance the body and coordination. Centralization is important, and to transmit. So you have the efferent pathway to transmit things out. And so you either have the somatic or the autonomic. Autonomic is the sympathetic and the parasympathetic. And the sympathetic is fight or flight. And so, but the parasympathetic vegetative, it's like the contractions of the GI tract. And so, but if you see a bear, a sympathetic is going to take over. But if not, just relax. The parasympathetic is going to allow you to digest your food. So here's the central nervous system, the peripheral nervous system, and the function of the different cells. You have the astrocytes, and it's associated with the blood-brain barrier. And then the satellite cells in the peripheral nervous system, and for ion exchange is what they're doing. Oligodendrocytes in the central nervous system correspond to the Schwann cells in the peripheral nervous system, and they produce myelin. And then the microglia, you have in the central nervous system phagocytosis. You don't have that in the case of the peripheral nervous system. So here we see the astrocytes, microglia, oligodendrocytes that are located in there. And in the peripheral nervous system, we see the Schwann cell. So this is one Schwann cell that has wrapped the myelin sheath around it. So this is the myelin sheath that's wrapped around it. That's what we see right here. And also that's what we see on either side of there. The gray thing in the middle is the axon, and then the myelin sheath, and then these are largely Schwann cell nuclei. Could have fibroblasts in there, could have endothelial cells and blood vessels in there as well. And so here we see the different shapes of the pseudo-unipolar is what it is, pseudo-unipolar neuron. That's what we have in the dorsal root ganglia. This is in the ear. We'll see one later. Now the structure and function, we have the pericardium, which is soma, or it's a cell body. It's a cell body, that's what it is. And then you have the axon helix right in through there, where the projection of the axon. Then you have the dendrites projecting up from the base. And then the axon is actually what connects the nerve cell body with the target tissue, except it synapses with it. It connects in a way, but it doesn't really touch it. There's a really little, there's a synapse space in between the two. Also, we have Nissl substance, Nissl bodies that we see right in through there. And you can see the granularity in these cells. You see that in the cell bodies. There's a nucleus, nucleolus. Here's a nucleus. And then the Nissl substance is the granularity that we see in through there. So if we look at the central nervous system up through there, we have the cell bodies, Nissl substance. You have oligodendrocytes making a myelin, and then when you get to the peripheral nervous system, you get the Schwann cells making the myelin. This is some peripheral collateral branches that comes out here, so it can have a motor end plate on the various muscle fibers. So this cell is telling these cells to contract, and so we can see the cell bodies. There's a nucleus, very euchromatic nucleus, nucleolus, then in the middle, and then we see the Nissl substance there. Here we see spines of the dendrites projecting in through there, interacting with axons from other cells. Axons of other cells would be a synapsin with the dendrite of this particular cell. And so if we look at Nissl substance, we can see the granularity there. We can see it right here. Also, if we look at electron microscopy, you can see it's really polyribosomes. And some of the polyribosomes could be free, or some of them could be on the rough endoplasmic reticulum. But basically, that's what we're seeing there when we see the ribosome-rich areas, which looks granularity, the Nissl bodies or Nissl substance that we see. Now, in terms of transmission from the cell body to the terminal, you have a couple of things. You have anterograde, which is toward the terminal, and kinesin is involved in that, and that's you got transmission in that direction. And then we have retrograde toward the body, and that toward the cell body, that is from the terminal out this way, and that's where you have dynin is involved, and that's where we get our tetanus toxin. That's where we get neurotoxic viruses, herpes, rabies. They follow this path to go back in and get into our brain. That's how they're able to take that path. But we also use that path, that same path, that it works with dynin to put peroxidase in through there, and it will go back in this region, and it will show you where the axon is in the target tissue. And by doing that, we're able to target which portions of the brain is associated with what portions of the body, because we can follow the nerve cells back into the brain. And so, like the head, this is the hand, the arm, the fingers. Area is a big area with the fingers, so you can really have agile fingers. Lips, very sensitive because they've got a lot of brain associated with the tongue. In through there, not so much with the pharynx. There's the tongue over here on this side, the jaw, lips, face, eye. And so these are different portions of the brain that's associated with axon terminals that terminate in the face or the eye or the eyebrow or someplace like through there. And we were able to do that by using the retrograde transmission of things from the terminal back into the location where the cells are located, where the cell bodies are located in the brain. Now, the function of myelin is increase the speed of the conduction, about 1 meter per second to about 120 meters per second. It's a high resistance, low capacity insulator is what it is. So this is the myelin sheath. This is the axon. Axon can vary in thickness, as you can see. Axon here, myelin sheath. And here we can see where the Schwann cell wraps around it. So it's really the myelin sheath is actually the cell membrane of the Schwann cell. That's what it is. Protects possible nutritional role, direct regeneration of axons, and we'll talk about that later on. So here we see the Schwann cell. There's this nucleus, and the Schwann cell cytoplasm that's wrapped around. The blue here is the axon here. So one Schwann cell can wrap around one axon. And so where one Schwann cell ends and another one begins, that's a node of Ranvier, as we see. Sometimes we have incomplete compaction of the cell membrane. That makes something, you can see a light structure there, the Smith-Landerman clefts. And these run at an angle, as opposed to this is perpendicular, that we can see. And neurokeratin substance is just neurokeratin substance that are located in the nerve cells. And then we see a Schwann cell with the myelin. This is it always have to have a double membrane that goes around it, and then here we can see the trilaminar structure of individual membranes. So it wraps around the axon. So the axon in the middle and the myelin sheath goes around through there. There's a node of Ranvier, which is the same as that. And then here we see the Smith-Landerman clefts, which is an incomplete compaction. Here we see the node of Ranvier where one Schwann cell ends and another one begins, and you see the swirling of the membrane, which is actually, you can see that right here. You can see the swirling of the membrane. You can see it right here, sorry. You can see it right here. So where one Schwann cell ends and another one begins, it's a node of Ranvier. So here we can see if you have a myeloid axon, then the axon is here. This is axon here, here, and here. It swirls around it. It can only swirl around one. It can't swirl around more than one. However, you had unmyelinated axons. They're just kind of like in the same sleeve as is your arm, so to speak. And so you don't have a complete circling of the multiple circling of the axon as you do. And here we can see this is a Schwann cell nucleus here, and you can see where several of the axons have been encased within the limits of the cell, even though it's not inside the cytoplasm of the cell itself. They just kind of tucked in its pocket, so to speak. So organization of peripheral nervous system, as I mentioned, you have ganglia. So you have things from the central nervous system telling the local tissue to get going. And so here we see in the digestive tract, we see longitudinal and cross-sectional smooth muscle, and in between the two, circular muscle, longitudinal muscle, and we see the ganglion cell. So this is a nucleus, nucleolus, and here we can see some satellite cells around these cells. Now remember, when we looked at connective tissue of muscle, we had the epimysium on the outside, the perimysium in the middle, and then in the very one around individual muscle fibers, you had the endomysium. You have a similar type thing in a nerve, except we call it perineurium is on the outside. And then the epineurium is on the outside. The perineurium is actually what's responsible for the blood peripheral nerve barrier. Is what it does. It has tight junctions between adjacent cells. Here you can see where a tight junction is on this perineurium fibroblasts around this Schwann cell and the axon that we see right there. So on the outside, epineurium, perineurium has major divisions, and also in the case of nerve, it has junctions, unlike muscle. And then the endoneurium is what touches individual cells. This is what touches the Schwann cells, is the endoneurium. So we can see that the epineurium on the outside, connective tissue, perineurium making the blood peripheral nerve barrier, and then you have the endoneurium, which actually is touching individual Schwann cells. So here we can see this would be like the epineurium and the perineurium around each one of these. So nerve has a very distinct perineurium around individual nerves, and then the endoneurium would be on the inside. Perineurium, again, endoneurium, epineurium is on the outside. Perineurium, you can see the fibroblast that goes around through there. These are myelinated axons, and here we can see the axon is kind of gray and the myelin sheath around on the outside. So you have sympathetic and parasympathetic as far as your autonomic nervous system, largely running the organs, the GI tract, wherever is the parasympathetic. But whenever you need to run, you're going to kick your sympathetic in. So nerve endings or synapses, efferent pathways we're looking at. Electrical ones, you have gap junctions, you have ion exchange channels, and then you have chemical ones, rapid plus negative, positive or negative responses. And the characteristic of synaptic vesicles, you got neurotransmitters there. You have a cleft, a motor end plate. This is what we're going to see. So here's a motor end plate in through there. So that this is muscle, and this is all part of the terminal in through there, where the nerve is telling the muscle to get active. Here we can see the motor end plates in through there, And so like this would be one of those, and you can see it's got the transmitters located in vesicles in there. You got the synaptic cleft in between. You got receptors on the postsynaptic surface that will respond to the chemicals that are in there. So this is a terminal one. This is a site of transfer. It could be a nerve cell, could be a muscle cell that we can see. And basically you get action potential comes down through here and through there, and then you have a release of the presynaptic vesicles into the synaptic cleft. And here we can see like there's this is a muscle cell in through there. This is the axon terminal, and this is a synaptic cleft right in through here. And so the vesicles would be transmitting so that the receptors on the muscle cell could respond. So the neuroactive substance, you got neurotransmitter, acetylcholine, for example. You got modulators and neurohormone like GABA, serotonin, widespread effects. So depending upon the effects you need, what kind of transmitters you have. And then here we see this is the innervation of the nerve. This is where the synaptic cleft is, and you can see in the vesicles of which they have actually released in through there. So this is on the nerve side of the synaptic cleft with the muscle. And here we can see it again. So this is the muscle, and this is the nerve, and you can see synaptic vesicles fusing with him and discharging their contents in through there. There's some coated vesicles and the recycling that occurs, but then, so this would be muscle cells, synaptic cleft. Vesicles transmitted. And so basically what happened is you got activation of the terminal by the action potential. So you get a nerve shock. You get the action potential come down through there. And so and then you have the opening of the calcium. So calcium influx causes the exocytosis of the product. So you get influx of calcium, exocytosis of the transmitter into the synaptic cleft, and then they interact with the receptors on the muscle cell or the cell that's being transmitted until you get a conformational change. And then you get depolarization. You depolarize the cell. Hyperpolarization makes it insensitive, and then you remove the transmitter. So you get the electrical shock comes down, calcium influx, exocytosis. Then you get the binding of the receptors to the transmitters. And then you get the cell to change the sodium transmission of that target cell, and that causes the action potential to be in that cell as well. So you get active potential coming down the terminal, open the calcium channels, influx of calcium causes exocytosis to occur, diffusion of the transmitters across the synaptic cleft, binding to the receptors of the target tissue, opening of their sodium channels, and that causes depolarization of the target cell. And then the transmitter is removed very quickly. So here we can see the ion channels in through there and how they change to open up the different channels. And also we have a recycling of the membrane that is run through here. You can see some membrane, the coated pit, we saw it a while ago, the coated vesicle, the coated pit, and then it comes back in here, and then they're actually reloaded. And so here we can see where you have exocytosis occurring in through there. Then these guys are pinching off of the vesicles, go back, bring in this is the coated vesicle that's coming back in through there. So you pick up the transmitter and you bring that back in through there, and that's reloaded for the next discharge to occur. So basically you get delivery from the initial substance that's down in through there, which ultimately goes to the Golgi, transmission through there, exocytosis to occur, and then you you have endocytosis occurring back to these vesicles, reloading of the transmitters, more transmitters put in, and then you get transmitter exocytosis again to occur. And so synapses can occur on axons, and sometimes they have different names depending upon where they are. Axonodendritic synapse on a spike, spine, I should say. And so here we can see synaptic vesicles here on to a dendrite, axon dendrite. And so there are different types of nerve endings, which are receptors. Some in neuromuscle spindles that we'll see. There's also presenting corpuscles, deep pressure or light pressure Meissner's corpuscles. And this one is a nerve ending. This is a muscle spindle, and you have a capsulated structure with some muscle cells inside there. And these are actually muscle cells. You can look and see the striations in them. And when they stretch, that tells a muscle how much it's stretched. As opposed to presenting corpuscles, we can see one here, and there's kind of like an onion. When it's squeezed, it causes action potential to occur in the nerve cell, in the nerve projection that's inside there. Then this is a Meissner's plexus. This is right up there on your skin. This is a light touch one that you can see these cells, whenever they're squeezed, they cause the action potential of the cell. You can see the Meissner's corpuscle looked kind of like a tornado that goes in through there. In the inner ear, we also have other types of receptors for listening to sound. You have receptors, of course, in the olfactory system that you see, the olfactory epithelium that smells. And so there's different ones: deep pressure, light pressure, just nerve endings, just free nerve endings that are in there. And these are the ones telling the central nervous system something, so it can use that information to decide what to do next. So here we see there presenting corpuscles in there, and the Meissner's corpuscle would be right up in there, but I don't really see one. And so this is the motor end plate that we talked about before, so that the nerve cells could interact with different muscle cells. Now also the myelin sheath is useful for regeneration of the axon. If you cut this thing and you still have the myelin sheath there, sometimes the cell can find that target. The axon can find that target cell again. So this was, and that's why over a period of time, sometimes you restore function whenever you have neurologic damage to occur. Other times, if it's damaged in such a way that it can't get there, then it won't return in several months later on. And so here we see body whorls. Look at these nerves in through there, going through there. Lots of big nerves that run in things. We want to thank the many original sources of some of these images that we've used and figures that we've used, and also some of the body whorl images. Next time we'll talk about blood, nerve, lymph system. Thank you.